Hi, this is Elisa Schwenk, and today we will be going over Nirenberg and Matthaei's and Nirenberg and Leder's approaches to determine genetic code. The main goal of these experiments was to determine which codons code for certain amino acids. First, let's begin with Nirenberg and Matthaei's experiment. For these experiments, Nirenberg and Matthaei began by combining a polycodon from an RNA, cell-free extract, and all the amino acids into a test tube. However, one of the amino acids was radioactive. If the protein that was synthesized was radioactive, then they knew that the codon coded for that specific amino acid. Let's imagine doing this with a poly-U codon. First, the poly-U, free cell extract, and all amino acids were added into a test tube. Next, they were incubated and translation occurred. Once translation was complete, a proton precipitate was formed. The protein was then filtered and checked for radioactivity. If the protein was radioactive, then the codon coded for the original amino acid that was labeled radioactive. After this was complete, Nirenberg and Matthaei discovered that poly-U coded for phenylalanine. They also completed this experiment 3 more times with poly-C, poly-A, and poly-G. While poly-G never synthesized any radioactive proteins, poly-C coded for proline and poly-A coded for lysine. Once this was complete, Nirenberg began another experiment with Leder. In this experiment, Nirenberg and Leder utilized a trinucleotide, which is a codon, a ribosome, and 20 charged tRNAs, one of which had a radioactive amino acid. But how was this experiment executed? First, codons were synthesized and added to a mixture of ribosomes and tRNA that were attached to amino acids, one of which was labeled radioactive. Next, the ribosome would bind the mRNA codon as well as the tRNA for which it specified. Finally, the mixture was passed through a nitrocellulose filter. tRNAs that were paired with ribosome-bound mRNA would stick to the filter, whereas unbound tRNA would pass through the filters. After this was done, the filter paper was observed to see if it was radioactive. If the filter was radioactive, then that codon coded for the amino acid that was labeled during the experiment. Through this experiment, Nirenberg and Leder were able to identify 61 codons. Through Nirenberg's experiments with both Matthaei and Leder, the coding dictionary for amino acids was able to begin forming for future scientists' use.